Welcome to today's episode of the Bio Bunch.
Today's topic will be ancient DNA and human history.
But what's ancient DNA?
DNA is deoxyribonucleic acid. It's like a set of instructions that ultimately determines everything about us. Ancient DNA is just DNA that's removed from ancient forms of life.
Scientists can look at ancient DNA to see the origins of life and our progression to humans. Scientists can use this specifically to see where humans came from and our common ancestors.
But how do we study DNA?
Let's consult our expert, Dr. Barrier. To do that, we're going to have to go to her lab. So let's go! Hi, Dr. Barrier. This is Dr. Barrier from the Genomics Lab in the museum.
Hi, would you like to learn about the micine?
I would.
So this here is one of our DNA sequencers. It allows us to actually read what the sequence of DNA is of different samples. One of the ways we use it is to look at the DNA of human samples and primate samples. And we can learn a lot from the primate DNA because we evolved from a common ancestor. And so we can learn from them, and it teaches us about ourselves.
Wow, that's super cool. Thank you so much.
Sure.
Thanks, Dr. Barrier. It is so important to note some of the legal, ethical, and social controversies that surround topics such as ancient DNA, human history, and evolution. Because social and ethical issues tend to be fairly similar, we will limit ethical issues to topics that involve religion.
All legal action regarding ancient DNA and human history stems from education and if it is legal to teach such topics. A prime example of this is a highly influential case of Tennessee versus Scopes. Ultimately, this court case decided that it is not legal for any public school to make the teachings of a religious document the standards for teaching in that public school. This meant that evolution could be taught and tested on regardless of whether the person's personal beliefs were of conflicting interests.
Another court case along these same lines is the court case of Epperson versus Arkansas. During this court case, which was brought against the state of Arkansas in 1968, it was decided that banning the teachings of evolution or the use of a book that teaches evolution is both a violation of the First Amendment freedom of speech and the Fourth Amendment, which ensures that all citizens of the United States receive the same protection of the law.
There's one more case that we want to talk about. The case of Kitzmiller versus Dover School System is yet another court case about evolution and creationism. In this case, fought in 2005, a school policy based towards creationism was ruled as a violation of the Establishment Clause, which prevents establishment of religion in America.
This same idea ties directly into ethical issues of ancient DNA and human history. The church and many people with deep religious beliefs trust their scripture that humans were created by God. On the flip side, scientists and some people in the community believe that we evolved into what we are today through millions of years. We are not here to simply tell you which view is right or wrong, but simply to put the findings of science into perspective.
Aside from the debate between creationism and evolution, there's not much of a social argument around ancient DNA and human history. This mainly is because if it is based off personal beliefs, it's not groundbreaking research that has new controversy other topics have.
Now, before we learn about early humans, we must start at the very beginnings of life. Let's go.
$3.8 billion years in the past, a storm is brewing right now as life begins to form on planet Earth.
Look, early forms of life are forming from a so-called primordial soup.
What is primordial soup?
Well, primordial soup is where life began. The primordial soup can be viewed as an ocean of organic molecules that came together to form the first life forms. RNA was most likely a precursor to DNA, as chemical groups were added and taken away. This evolution of molecules is fascinating. In my hands, RNA is becoming DNA and forming biology's first life form, bacteria. Ribonucleic acid is losing an oxygen and a hydrogen to form uDNA, or uracil DNA. Oh look! Now the uracil bases are having chemical groups added to them called methyl groups. They're thymine now. From this point originates all forms of life. bacteria continued to evolve into new organisms that were both single-celled and multicellular. Now, back to the lab.
Now, in order to talk about the development of multicellular organisms, we have to discuss endosymbiotic theory.
But what's endosymbiotic theory?
Endosymbiotic theory states that a eukaryotic organism originated when an archaebacterium merged with a eubacterium to form a single cell.
This led to cells having mitochondria, which facilitates cell respiration, and then chloroplasts, which facilitates photosynthesis. Now, let's watch a short animation. The evidence for this theory lies in the fact that both mitochondria and chloroplasts have their own DNA.
This shows that at one point, mitochondria and chloroplasts existed by themselves before merging into other cells.
The importance of this is that scientists can track mitochondrial DNA back through our ancestors. We will talk more about this later with the theory of evolution. Now let's run into some early humans.
Now, it's about time that we get to some early humans. But before we can talk about them, we have to mention Luca.
What's Luca?
Luca is the last universal common ancestor. From this point, every current living species diverged. Now, let's start discussing our ancient ancestors.
How are we going to do that without any pictures?
Luckily, I have some in my back pocket.
Australopithecus is the earliest known genus of hominids living from 4 million to 3 million years ago. They lived in the eastern region of Africa and did walk in an upright manner similar to modern humans, but their skull was much different from ours, looking more like that of great apes. Genetically, Australopithecus was extremely important for human evolution, with studies showing duplications of the SRGAP2 gene between Australopithecus and the genus Homo. SRGAP2 controls how neurons grow. The next genus of hominids that are most similar to modern humans is Homo. We are a part of the Homo genus. Within this genus, there have been many species that have ultimately led to us, Homo sapiens sapiens. But the first for this genus was called Homo habilis. Homo habilis, or handyman, occurred just before Homo erectus and is the oldest known member of the Homo genus to date, living from 2.4 to 1.5 million years ago in eastern and sub-Saharan Africa. The only distinguishable difference between Australopithecus and Homo habilis is the structure of the face and brain cavity being more similar in Homo habilis to modern humans. Now let's see a picture of these side by side. As you can see, the brain cavity of Australopithecus is much smaller than that of Homo habilis. It is also clear that Australopithecus has much more distinct eye sockets than Homo habilis, making them very easy to tell apart. The next in our series of ancestors is Homo erectus, which lived from 1.6 million to 200,000 years ago, and spread far and wide across southern Africa, eastern Asia, and southern Europe. Repeating our common pattern, the most noticeable difference between Homo erectus and Homo habilis is the structure of the head. However, it is also important to note that they are the most skilled tool makers to date and already knew how to make and use fire. Here are some tools that Homo erectus used. They're pretty much pointy rocks. Let's compare Homo habilis and Homo erectus side by side one more time. Now these two are very difficult to tell apart, but let me point out a subtle difference. Notice that the jaw of Homo habilis protrudes slightly and their teeth are more forward than that of Homo erectus, whose teeth are straight up and down. This is virtually the only identical difference in the two species, minus looking at their genome and overall body structure. Now, Neanderthals are the most clearly distinguishable early human thus far. Neanderthals lived between 600,000 and 200,000 years ago between Africa and Eurasia, being as far north as current Great Britain. They had an enormous head with brains even bigger than our own. They were also stockier than humans, as you can see here. Though it is thought that we are not directly descended from this species, we do share some amount of DNA that makes scientists believe that some of point, our ancestors interbred. Scientists have completely sequenced both of the mitochondria and nuclear DNA of Neanderthals. This was called the Neanderthal Genome Project and was completed in 2010. Look at this side-by-side view of Homo sapiens and Homo neanderthalis. You can see that Neanderthalis is much shorter than Homo sapiens and also a lot more stocky. If you look at their brains, look how much farther back the brain cavity of Homo neanderthalis goes. You can definitely see the larger brain cavity. You can also see the flatter forehead in the Homo neanderthalis versus the Homo sapiens. The term Homo sapiens is used to describe hominids with the same anatomy as us. Homo sapiens lived from 200,000 to 60,000 years ago, primarily in Africa and the Middle East.
Before we go, it is important to address some of the ethical and scientific controversy that surrounds Neanderthals.
Through comparisons of Neanderthal DNA and our DNA, some scientists have discovered that modern humans possess 1 to 4% of Neanderthal DNA. It is important to note that the controversy here lies in how we came to possess this DNA.
Some scientists believe that modern humans and Neanderthals interbred and that modern humans outcompeted or simply killed Neanderthals. Other scientists believe that Neanderthal DNA was simply absorbed into the modern human genome because of better fitness to the environment and larger population size.
Another important point of scientific controversy is the technological difficulty in sequencing the ancient DNA. Most ancient specimens often have DNA that has been degraded or contaminated over time, leading to complications in determining similarities between modern humans and ancient species.
Now that we have looked at some older forms of humans, we need to look at how we are still evolving. For this, we have to go back to the lab.
Let's go!
It is believed that we are still evolving today, and there is controversy over whether or not we're influencing our evolution due to our newly discovered understanding of genetics.
Scientists have lots of questions about our evolution.
Are we affecting evolution, or is this simply a new form of evolution?
Will selecting against specific traits via in vitro fertilization cause our species to head in a new direction?
Is it ethical for us to be selecting against specific traits?
How will new laws affect research into new areas of genetics?
On the legal issues of current human evolution, in vitro fertilization and selection against specific strands of DNA to avoid undesirable traits is currently legal in the US. However, a more imminent issue is gene line editing.
Gene line editing is still in development, so it is not at the absolute forefront of legal action, with few laws pertaining specifically to gene line editing. Recently, laws making human modification both easier and harder have been passed. In 2015, a bill was passed to prevent the modifications of human embryos from receiving federal funding. However, in 2016, clinical trials for CRISPR gene editing therapies were approved.
Now, let's move on to some more ethical issues. Similar to earlier, we will make ethical issues topics of opposition between the church and scientists.
In most of these cases, churches and people with deep religious beliefs believe that creation is the job of the Creator and that us altering the sacred human genome would be sinful.
Scientists, on the other hand, look at this technology as a medium in which we can learn. Most scientists wish to explore the possibilities of individualized genetic treatment for people, and possibly as a way to prolonging the lifetime of humans.
Now it's time to look at issues that are more on the social side.
It's important to note the argument that any new developments in this technology will only be available to the wealthy 1% and powerful people of our world. With this, many people counter that many countries have a system for redistribution of opportunity, and even though this does not work perfectly, it is better than doing nothing.
The end-all argument against this research is whether or not we are creating a weapon to be used against us in the future by editing the human genome. Many people wonder how we can defend ourselves.
Ultimately, these are all questions that must be addressed in the near future by our species. But, in the meantime, it is most important to educate yourself on the topics discussed in today's episode so that you have the chance to form your own opinions. Wow, that got pretty deep, but that's all we have for today.
Next time, catch us on our talk about the modification of histones through methylation, acetylation, and phosphorylation.
Now to play us out, our musical guests Tess Cross and DJ DNA.
Genetics, for you and me.
Come on, guys. ATCG.
Nucleic acids are part of us.
That's proven, man. Don't make a fuss.
What the stuff does is a little complex.
But without it, we'd be a mess.
We've come a long way over time.
All right. Just listen to our rhyme.
Australopithecus is the earliest one.
Lived at 3.2 million, then Lucy was done.
Next came the Homo species, after all.
Habilis, Erectus, and Neanderthals.
We are sapiens. Guess we're wise guys.
We came a long way, ain't no lie. You may not believe me, and that's okay.
Each different person thinks in a different way.
What's important to know is how everyone thinks about all the various genetic links.